So you start with Morgan, proving that the inheritable material, the instructions that it's passed generation to generation, carried on chromosomes. Griffith, showing that it can be passed. And if it's passed from one cell to another, then a new cell can be transformed. Avery McLeod McCarthy, showing that what Morgan was looking at, what Griffith was examining, was the DNA. The DNA is what carries the information. The problem was that even back then, back in the 40s, late 40s, going into the 50s, the argument still stood that it couldn't be the DNA. The DNA wasn't complex enough. The DNA just didn't have enough going on for it. The argument against Morgan was his fruit flies were too complex. You can't control all the variables. Problem with Griffith was he was dealing with bacterial cells that still had too much going on. The problem with Avery McLeod McCarthy was there's still too complex of things going on. We don't know exactly what all could not be there. The bacterial cells are still too complex. So, if bacterial cells are complex, let's go to the most simplistic organisms on this planet. Viruses. As you see here on the right, those lunar lander spaceship looking things are viruses. Specifically, they are what are referred to as bacteriophages. Phage, small particle, what we now think of as virus. Bacterio, meaning it's a virus that can only infect bacteria. What's interesting about bacteriophages is there's only two components: proteins and DNA. Inside the phage head contains the DNA. Everything else you see here, the phage head, the tails, the tail fibers, all of this, that's different proteins. All told, I think there's like 12 different types of proteins come together to make this. There you go. DNA proteins. No lipids, no RNAs, no anything else that could carry information. Early 50s. The research group under Hershey and Chase decided to examine these bacteriophages, looking for the final proof. Is it protein or is it DNA? Because when you look at chromosomes, that's all chromosomes are. They're protein and DNA. The counter arguments going back and forth at this time was that DNA is not complex enough. Proteins, with all the different amino acids, the different sequences of amino acids, and the different things you could add and subtract from amino acids, is a very complex language. The DNA that's there in the chromosomes is just there to hold the proteins together. The other argument is it's the proteins that are just there to hold the DNA together. So back and forth, in comes Hershey and Chase. They chose bacteriophages, specifically the bacteriophages known as T2, only two components, DNA, protein. What they knew about viruses at the time was one virus has to get into a cell. Once that virus is in the cell, the cell gets hijacked, becomes a virus-making machine. So, T2, E. coli. Only two things you need for this experiment. E. coli is just going to be a host, okay? T2 is what's going to infect the E. coli. The experiment is to examine what gets inside the cell. Does the protein get inside the E. coli cell, thus bringing in the information? Or is it the DNA? Does the DNA have to get inside the cell, thus bringing in the blueprint? So they designed this experiment, going to look at two forms of the T2 virus. The first form was T2 virus that was cultivated in the presence of radioactive sulfur. Sulfur is going to be there for the disulfide bridges as part of the amino acids that make up the proteins of the virus. Sulfur does not occur in DNA. The other batch is going to be grown in the presence of radioactive phosphorus. Okay, think back to our earlier chapters when we were talking DNA nucleotides. Okay, the sugar phosphate backbone. Okay, each nucleotide, to be a complete nucleotide and non-nucleoside, has to have the phosphate. Phosphate does not occur in any amino acids. So, here in the pink, radioactive proteins. Here with the blue, radioactive DNA. They isolated the two forms of the radioactive viruses. Remember, they kept them separate. They isolate them, so they now have two separate solutions. They allowed them to incubate with E. coli, and then they put that solution in a Waring blender. Yeah, blender, just like you would make margaritas with. Turns out, yeah, blenders are a scientific tool. The spinning of the blades created such shear forces that whatever was on the outside of the cell would literally get sheared off, pulled off. They collected the solutions from the blenders and they centrifuged them. Now, they centrifuged them in such a way that the bacterial cells, which the Waring blender is not going to break apart, will settle out. Anything not inside the cells will still be up in the solution. Then they examined where the radiation was. For the radioactive protein solution, what they found is that the bacterial cell pellet was not radioactive. All the radioactivity was still up in the solution. So radioactive proteins did not get inside the cell, which is counter to the radioactive DNA solution. When they examined the solution, the supernatant above the pellet, nothing. No radioactivity present. All the radioactivity was down in the bacterial cell pellet, which told them the radioactive phosphorus was now inside the E. coli cells. If the radioactive phosphorus is inside the E. coli cells, that means the DNA is inside the cells. The only way that could happen is if the DNA is the component of the virus that carries the blueprint on how to make new viruses, and thus that is the component that has to get inside the cells. Ding, 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 ding. Final. Proof positive. Morgan's work, too complex. Fruit flies, even though he did show the chromosome, but not what part of the chromosome is actually doing the work of carrying information generation to generation. Griffith's work, too complex, even with the bacterial cells. Don't know which component of the dead S is being used to transform R. Avery, McLeod, McCarthy were able to show that it's DNS, but hey, there's still too many variables. Bacterial cells, still too complex. Can't prove it beyond a shadow of a doubt. Enter in Hershey Chase. Hershey and Chase were able to finally prove, using the most simplistic organisms on the planet, that it's not protein, but it's DNA that carries the blueprint, what we now think of as the genes.